Hello everybody, thank you for coming. Today I'd like to talk about some of the theoretical work that I've been doing in genetic architecture. I'm specifically interested in how the genotype is connected to the phenotype and about pleiotropy. So pleiotropy is when one locus or gene affects more than one trait. It is important because pleiotropy is thought to be very common throughout genomes, with most genes having some degree of pleiotropy, and also because many traits are thought to be constrained by pleiotropy by being connected to other traits. Now, in the past, patterns of pleiotropy were thought to be nearly universal. That was the implicit assumption. But more recently, empirical data has shown that there is a highly structured modular structure to pleiotropy. And this is the importance of that is that sets of traits in modules can evolve independently from other sets of traits. Now I'm going to be talking about a specific type of pleiotropy, and that's mutational effect pleiotropy. And that's when a mutation at a particular gene or locus has an effect on more than one trait. You can think of that mutational effect as a distribution that is centered on zero, where a mutational effect that is close to zero is much more probable than a mutational effect that is far away from zero. And that mutational distribution could be circular, like the one you're seeing here, or it could have some other kind of shape. If there is mutational effect correlation, that means that you have a mutational effect that in this case has a positive effect on one trait is more likely to have a positive effect on other traits. Here the example is a positive mutational effect correlation of 0.5, and you can see that there's this elliptical shape of the distribution. Now I want to mention that mutational effect correlation is the only way that pleiotropy can actually have an effect, the only way pleiotropy can lead to genetic correlation and phenotypic correlation. In evolutionary quantitative genetics, one of the tools we use is the G matrix, the genetic additive variance and covariance matrix. And what that represents a matrix with the components that has the variance for each trait and the covariance between any two traits. Here the example is for 2 traits, but of course it could be for more than that. And what we can do with this tool is we can apply an eigen decomposition to it and get some idea of the dimensionality of additive genetic variance in trait space. I'll give you an idea of what that means. So here when we do a decomposition of a G matrix with 2 traits, what we get is a couple of things. First, it decomposes into this eigenvector matrix, which gives us 2 eigenvectors that describe the orthogonal axes under which genetic variance lies. Here they're labeled as eigenvector 1 and eigenvector 2, but they can also be principal component 1 and principal component 2 if you are more familiar with principal component analysis. And then with each of those eigenvectors there's associated eigenvalue, and that determines the magnitude of that genetic variance, where eigenvector 1 will have the largest amount of genetic variance and will be the axis of least resistance to selection. So here in this population, we have each of these dots is an individual and their additive genetic breeding values for these 2 traits. The G matrix itself is this elliptical shape that circles about 95% of that population, and you can see that it has this elliptical shape which shows that it has genetic correlations between these 2 traits. The usefulness of this G matrix comes from its ability to predict the response to selection. So if we have a genetic variance covariance matrix, and we know the direction and strength of selection for a set of traits, here represented by this beta vector, then we can determine what the response to selection will be. In this example that I'm giving, the beta vector is selecting for greater values on trait 2. line of least resistance is this eigenvector 1, the longest axis of the G matrix, and our response to that selection will be somewhere in between the two. Now I'm going to put these ideas together of genetic architecture into this evolutionary quantitative genetics framework to answer a few questions. The first question is whether the eigendecomposition of the G matrix can tell us something about the genetic architecture. And the way I went about this was designing some different genetic architectures. Here's 5 different genetic architectures. Each one has 120 loci and 12 traits, but they're divided into a different number of modules. In the first one, with modularity 1, we have 120 loci, and they all affect all 12 traits. This is the universal pleiotropy that I was speaking of before. And that goes all the way down to modularity 6, where we have 6 modules, and in each one of those modules we have 20 loci affecting 2 traits. I took these 5 genetic architectures, and I put them into 5,000 individuals and simulated them in the individual-based modeling population genetics simulator NEMO. I exposed them to 10,000 generations of stabilizing selection on the traits, on those 12 traits, followed by 2,000 generations of directional selection either on one module or on one trait. I did this with and without mutational correlation to see what the effect was, and I did several replicates. Now, when we look at some of the results here, we have the average eigenvalues of those G matrices after 10,000 generations of stabilizing selection. And what we can see is when we have mutational correlation, that's this graph on the left, when a set of traits are in the same module, that they have shared genetic correlation, and that stems from the mutational correlation from pleiotropic. And when that occurs, what we can see is that, for example, with modularity 1, all of the traits are in one module, and we have one eigenvalue that stands out above the rest. When we have 2 modules, we have 2 eigenvalues that stand out above the rest, and all the way down to modularity 6. The same is not true when we have no mutational correlation in our simulations. So, what we can say is that the genotype-phenotype modules are detectable in decomposition of the G matrix, but only when there is mutational correlation. And I want to emphasize that we found similar results when we tested this with different population sizes, with correlational selection included, overlapping modules, and when we scaled the mutational effect size. The second question that I wanted to ask was how genetic architecture affects the response of a trait to selection. Here we have the results after the 2,000 generations of directional selection, either on one trait or on one module. And we can see that the populations are evolving from the origin to a new optimum with the arrows every 10 generations. Now when when directional selection is on one module, the axis of least resistance of the G matrix is in line with the beta vector, and they move directly towards the new optimum. Whereas when we select on one trait and there is mutational correlation, then the direction of the beta vector is not in the same direction, and we get this deviation. Here. What we can also do is we can look at how many generations it actually takes for them to cross the same threshold. Here represented by these large circles. When we do that, first looking at just selection on one module, what we can see is that with mutational correlation, larger modules, so modules with more traits in them, take less time to evolve to the same fitness threshold, whereas smaller modules with less traits in them take more time. The opposite is true when we have no mutational correlation. When we have no mutational correlation, these large modules take a lot longer to evolve to the same fitness threshold. And this gives us an idea that the number of traits in a module has some effect on the response to selection. When we compare that to selection on one trait, what we find is the opposite trend. Mutational correlation will slow down larger modules to a greater degree than it will smaller modules. This gives us an idea of this, how mutational correlation can affect the response to selection. So what have we learned? Well, we learned that two things about genetic architecture can affect how the response to selection in a population. One is the mutational correlation, where larger modules benefit from mutational correlation, but only when selection is on a whole module, and the opposite when selection is on one trait. And the second thing is that the number of traits in a module will affect how fast the response is to selection. And which one of these is more important or a more effective target of selection for response time to adaptation, we're not really sure yet. Right. With that, I'd like to thank the Guillaume Lab and my funders at the SNF, and I'll take questions. No questions? All right. I guess it was perfectly understood. Thank you.